large scale symposium. Then in 2018 also, and after that COVID came, so all the activities closed. So then after that, we are reviving it this year, right? So the idea is that you get idea, you get some awareness about what is immunology and how it is applicable to different aspects of human life, particularly genetic disease and other immunogenetic diseases. So with this, now I request Dr. Himanshu to please carry forward this program. Thank you, Dr.
Thank you, Dr. for such a nice and elaborate introduction. एक बात बोलना चाहता हूँ, बिटवीन देन एंड नाउ जब मैंने आपके लेक्चर्स लिए होंगे और अब what advancement has taken place in PGIMS Rohtak, that is we are now in a position to do the human genome sequencing in our department. So this was not possible that time, and during as you might have known, the Human Genome Project got completed much earlier than the expected time. So there were technological advancements. And technological advancement has been so enormous कि आज वो machine इतनी छोटी हो गई है कि आप अपने hand में रख सकते हैं. It is just like a stapler. So that is the power of technology nowadays. And all these sciences are kind of interlinked. ठीक है आप ह्यूमन जीनोम पढ़ रहे हो या you are looking at human genome, but within those genes are also your immunity genes that are being transcribed from there. So all these sciences are interlinked, and that is why it forms a multidisciplinary or a transdisciplinary subjects nowadays. So basically it is good to jump from one subject to another, and it is possible only by molecular studies or molecular medicine. Whatever skills you might have today, if you have that commitment and that zeal of learning, if you can express that, you tell your policymakers that this is the course you want to pursue, it is possible that you can jump even from courses to courses, and you can to come to hardcore molecular medicine or immunology field or genetics field or whatever. But उतनी लगन और dedication होना चाहिए कि you can spend that much time. And basically, dedication वो होता है जिसमें आप रात और दिन अपना लगा सकते हो. So at this age of time, you are uh, basically you are kind of alone. You don't have a family. After some 5 years from now, many of you will get married. The things will be much different that time. So that is why it's a matter of time. You have to get uh, take decisions as soon as possible and do the right work. So in very soon we will be starting once our collaborators. Now all the lectures today are at least we have 3 lectures, so those will be from different cities. First lecture is by Dr. Pratapchan Mali. He is in Jaipur. The second lecture will be from New Delhi. The third lecture will be from Mumbai. As we go through the course, I will keep you walking or introducing you from time to time. So, and as I said, this is for the first time that whole world is will be watching you. Okay, so this program is live, and people from many countries will be watching us. And these immunological societies, they take a small time because they have to uh, you have to apply first and get their sanctions. They also give a small amount of money for celebration. काफी चीजों में खर्चा होता है कि, so all those things are covered basically, and they want to see how the function is going. So 10 a.m. onwards, we will be absolutely live, and there will be people from different places who will be watching us, and we will be also watching some of the people. दीपक होगा मेरे ख्याल से, दीपू बोल, उसको बुलाना जो ये मैनेज करता है। उधर बाहर गया था मेरे ख्याल से, उधर से नीचे चला गया, कुछ पता नहीं। ये रिमोट देना सफेद वाला। कोई स्विच ऑफ है क्या? चल गया। इसको जरा set करो। ठीक है, तो एक बार लाइव आ जाते हैं, सबसे छोटा फिर करते हैं। हाँ सर, मैंने आपको लिंक भेजा था व्हाट्सएप पे नहीं था, मैंने ईमेल पे भेजा था। व्हाट्सएप पे भेजूं? चलो मैं अभी अभी कर रहा हूँ। ठीक, ओके। क्या शक्ति? कौन थे? वो क्लास छूटी थी अभी, चले गए वो। और फर्स्ट ईयर वाले आ रहे हैं क्या? नहीं तो उनकी तो क्लास होगी ना फिर, फर्स्ट ईयर वालों की। सेकंड ईयर वाले अगर जा रहे थे तो उनको बुला लेते। देख लो जो जो कुछ हो सकता है। Good morning, Dr. Pratap. Okay, can you hear me? Bluetooth speaker on करना, connect the Bluetooth. Just wait, we are connecting the speaker. हाँ, यहाँ रखो, उस तरफ. So, is the speaker connected? Boat Stone 1000 connected. Hello, hello. Hello. इसमें साउंड किधर से बढ़ती है? सर कुछ थोड़ी सी बात कीजिए.
हेलो.
हाँ जी, हाँ जी.
हेलो.
यस, वेरी नाइस.
आ रही है सर?
यस, परफेक्ट. So, very good morning to you, Dr. Pratap. इसको स्क्रीन को आप थोड़ा सा नीचे कीजिए तो पूरा विजिबल हो जाएगा. ये उसमें क्यों नहीं आ रहा? This screen has to come there. ब्लूटूथ से है ना? ब्लूटूथ है। एक मिनट सर, लेट अस हम कनेक्टिविटी पूरी करते हैं। अरे और इससे निकलो बाहर, क्या कर दिया, निकलो इससे बाहर, इधर इसको क्लिक करो, कंट्रोल पी कर, ये पी करना था, ये हो गया डुप्लीकेट। चलो आगे उधर। अब ये क्या कर दिया, इसको इससे बाहर निकलो। ओके सर, परफेक्ट। So this is Dr. Pratap Chand Mali. He is professor in Department of Zoology, Rajasthan University, Jaipur, and he had his PhD with Dr. VP Singh, I believe, sir. So he was the last PhD student of Dr. VP Singh. VP Dikshit, yes. So, and his work was entirely on anti-fertility uh, in because that time, since 1980s, that was a time when population explosion of India was taking place, and there were lot of effort, even in TV, Doordarshan, all the time there were many campaigns to reduce the population of India or to constrain. हम दो हमारे दो उस समय सब जगह सारी दीवारों पे ये ऐड दिखते थे, तो ये तब उस जमाने की बात है। एंड उसके बाद फिर काफी रिसर्च की गई, एंड वन ऑफ द बिगेस्ट रिसर्च दैट वाज डन इन इंडिया was by Professor G.P. Talwar, who made the National Institute of Immunology receiving crores and crores of grants to make a anti-fertility vaccine for India, because that was the thought that that time कि ये कुछ मेडिसिन साइड से ही कोई इसका निष्कर्ष निकले, because many other devices were in use, लेकिन population control नहीं हो पा रही थी. So that is why Dr. G.P. Talwar, who is also known as the father of immunology in India, he was assigned the work of building National Institute of Immunology, and after, with that, there were many other researchers who were trying, just as Dr. V.P. Dikshit was also trying, and उनके शिष्य जो डॉक्टर प्रताप माली, जिनका हमारा सौभाग्य है उनके साथ बात करने का, he has also worked in field of anti-infertility. He has presented his work in many international conferences. He has visited very well traveled worldwide. विद्या ही विदेश गमनी, जितनी आप विद्या लोगे तभी आपकी आपको पूछा जाएगा जग में, और आप लोग को सब सब लोग बुलाएंगे। तो ये वो हस्ती है जिसको सुनने का आज हमें सौभाग्य मिला है। और let us let me show you your our audience, sir. एक बार इसको साइड में करता हूँ, so you can see our audience. Yes, nice. Yeah. So these are our students from BSc Nursing and MBBS or Physiotherapy, Falana Dekana, because ours is a Pandit B.D. Sharma University of Health Sciences. So we overall look at, besides MBBS, we look at several paraclinical courses also. So सभी field से बच्चे आते हैं, and these are our own students. So with that, we will start the inauguration function, sir. And may I request Dr. Dhara and other faculty to come on stage. Also, Dr. Shakti Singh, please join us.
Thank you, Dr. Himanshu, for this introduction.
You are very welcome, sir. Sijja? Yeah, take picture of the people. Sir, you just consider that you are here.
Yes,
So,
I'm feeling like it.
yeah, एक दीपक आपकी तरफ से मैं भी जलाऊंगा.
Yes.
आप लोगों में से कोई सरस्वती बनना जानता है? जो है जल्दी आ जाए इधर. Come on the stage, please. माइक की जरूरत नहीं है, वो सीधा स्पीकर है ना, तो वो कैच करता है.
शारदे माँ हे शारदे माँ हे शारदे माँ हे शारदे माँ अज्ञानता से हमें तार दे माँ हे शारदे माँ हे शारदे माँ तू स्वर की देवी है संगीत तुझसे है हर शब्द तेरा है हर गीत तुझसे हम हैं अकेले हम हैं अधूरे तेरी शरण में हमें प्यार दे माँ शारदे माँ शारदे माँ शारदे माँ शारदे माँ मुनियों ने समझी है गुणियों ने जानी वेदों की भाषा पुराणों की वाणी हम भी तो समझे हम भी तो जाने विद्या का हमको अधिकार दे माँ शारदे माँ शारदे माँ शारदे माँ शारदे माँ
Please be seated, everybody. Thank you so much. That was very melodious और काफी उसमें क्या बोलते हैं पढ़ने लिखने की भावना थी. तो इसके साथ आई विल रिक्वेस्ट डॉक्टर प्रताप टू स्टार्ट हिज टॉक. He will be talking on immunocontraception as I have given you the introduction. अच्छा वो उनकी फोटो खींचिए. Sir, you can share your screen and start.
Yes. Now you are. Hello.
Yes, hello, sir.
ये दिख रही है ना स्क्रीन पे पीपीटी.
Yeah, I can see. Make it full screen,
Okay.
sir.
Hello.
Sir, please make it full screen.
Full screen. Okay.
Yeah.
It is.
Very
now it is
nice.
better.
Perfect, sir. Perfect.
Thank you, Dr. Himanshu Raikwa, for inviting me to deliver my talk on the day of immunology, International Day of Immunology. Now I am going to start my talk. As you know, about each and everybody know about the contraception, and I think all until reproductive age need contraception somewhere they stay any time. And what is contraception? Contraception is a way to prevent the pregnancy. You can say the birth controlling method. Right? there are different types of contraception, and some are more effective than others. Contraception is a method, or you can say artificial method or technique, mainly used to prevent pregnancy as a consequence of sexual intercourse. And when a sperm, as you know that when a sperm reaches the ova in the woman, she may become pregnant. And contraception is a method that prevents the phenomena of conception by stopping egg production, keeping the egg distinct from the sperms, or by stopping the fertilization mechanism. How the contraception method works? There are many different types of contraception options, although different contraception you can see here in the picture different type of contraception. The barrier that is use a barrier method unknown in injectable oral contraceptives, implants, and other property and other methods. They are different type of contraception methods, and they are used for the to control birth rates. As you know that nowadays India lost the China population, and now we are the most populous country in the world. So it is, you know that all things are related to the populations, who are especially human populations. So to check the population or to control the birth rates, use of contraception is essential. This is the method when the mechanism of fertilization and the pregnancy occurs. As you know that in males, hypothalamus produce GnRH, and GnRH activate or stimulate pituitary to produce FSH, LH, and in females it is stimulate FSH in ovary to produce oocyte, or we can say egg or ovum. And male, it is induce or stimulate the production of sperms, and then they both gametes are fused to form a zygote. After that, mitotic serial mitotic divisions start and to form the blastocyst, and then blastocyst is implant inside the uterus or endometrium of the uterus. So contraception has come a long way from when the first oral contraception approved for the use by the FDA in 1960. As you know that this FDA in 1960 approval the use of contraception, India launched family planning uh, at the national level at the, by the government, Indian government start the use of contraceptive or family planning methods in 1952. The contraceptive choice available to women at this time include steroid, contraceptive, uterine devices, barrier methods, spermicidal compounds, natural family planning method, male and female sterilization, and recently available emergency contraception. So, undoubtedly, there is a call for a better method of contraception. As you know that there are several types of contraceptive method available in market to use effectively. However, they have some kind of drawbacks, or you can say side effects. About 48 to 48.2% of the couples practicing family planning methods in India. And how, although where India is the overpopulated country, and first now in the first country in the world where the population. Overpopulation has been overwhelming drive with limited resources for the country. The government of India adopted National Family Planning Program for development of newer technology in this field. Now, the Department of Biotechnology and Indian Council of Medical Research since the mid 70s are trying to develop birth control vaccines which would be effective for both sexes. That's, despite high expectations of safer, effective, economical, longer acting contraceptives, today there are no licensed contraceptive vaccine available in market. It is fact that our long journey started from this use of the contraceptive methods or the to check the birth control. But now still there is no 100% effective and safe method available for the use of couples. Nevertheless, the role of vaccine undoubtedly exists as an aid of birth spacing as as a non-surgical method of generating sterility. As you can say that there, you can divide it all the method, all the types of contraceptive methods. Yeah, you can say this timing method, or you can say spacing method, or you can say another one is the permanent method. The permanent method include vasectomy in males and female overectomy. Indian scientists have made not but the conclusion in exploring the possibility to develop an effective and safe contraceptive vaccine as earlier Dr. Himanshu mentioned that India has a significant contribution in the development of safe contraceptive vaccine and achieving contraception by means of a vaccine is the original approach which demands initiation of a specific antibody response against antigens critically involved in the process of mammalian reproduction. This and beside the emerging contraceptive methods for humans, there is an urgent need of novel contraceptive methods for fertilization in domestic animals. As you know that there are several species, or you can see the street dogs in local areas, or you can say in the rural areas. So it is also essential or necessary to control their population. So for the contested methods also used for the control to the control of population of wildlife like wild horses, deer, elephants, stray dogs, and cats, zoo animals, and cattle. The non-surgical human method is a less expensive and long-lasting is required for controlling farm, feral, stray, and domestic animal population. Immunological approach to block fertility species has been found to most suitable modality and down considered attention lately. As you know that there are several methods being used for the control of population for the wildlife and other animals, and most suitable it is or easily it is the ecological approach to control fertility or to block fertility. Wildlife conservation has its own set of unique challenges not prepared for human contraception. So, what can we can do? The methods of the annual contraceptives. Do all methods of contraception work as well as another? As it is imagined that all methods or contraceptives work in the same way, or they may be safer. But we can say that there are several type of contraceptives which have some kind of drawbacks or side effects. Some form contraception are more effective than others. As you know that in females there are several types of contraceptive methods. They use in the daily as a oral pill. They are made by the steroid hormones, especially estrogen and progesterone. But they have some kind of side effects like weight gain, abnormal bleeding, and some may cause cancer and CSD. Some particularly long-acting and long-acting reversible contraception are more than 99% effective at preventing pregnancy. Contraception is effective as possible. For instance, taking a hormonal contraception every day without a monthly break. How do you know which method should be used? Different methods suit different people. As you know that if couples have completed their family, so they can use a permanent type of contraceptive methods to permanently block the fertilization process or other process. So and their other people they not complete their family or they want to avoid the unwanted pregnancy, they have to use some kind of temporary contraceptive methods. You can say this is a various methods or some kind of another copper T or injectable contraceptives. Your choice of contraception may change depending on for your age. So the choice depend on the age and your health and status of your relationship. The nurse or doctor will ask you about the health or family. So, if any persons who wish to use contraceptives may consult with trained nurse or doctors about their contraceptive kind of contraceptive have to use. How old do you have it to get contraception? So you can also ask about the age at the particular age, have to be used to contraceptive or not. People of any age can get contraception from the nurse or doctor. And yeah, what are the different types of birth control? You can see here the picture, different kinds of contraceptive methods and which are using worldwide. They use it for the to check or block the fertilization or other process. If there are various methods, then for the male, there are male condom, for the female condom, and contraceptive sponge specimen sites that kill the sponge cells and diaphragm and cervical they can stop or block the passage of fertilizer. And oral contraceptives they may be used by women takes every day. They may gain progesterones or progesterone and students. And contraceptive patch, the patch that a woman puts on her skin each week, the patch releases hormones into the bloodstream. And vaginal rings, injectable birth controls, and injection of the hormone that a woman or man that can get every month or 3 months, or you can nowadays there are 3 years gap injections are available for the use. And implantation, a single thin rod that provides insert under the skin of woman's upper arm. It is the purpose. So, and other that is the stop for spacing methods and include the interdependent devices. A small T-shaped device. They also call copper IUDs or other metals are used, and along with this they also use hormonal. So they are like combined type of contraceptives. A small T-shaped device that a provider insert the interest. This is done by the provider office. UID IUDs last for 3 to 10 years. So you can have you can prevent the pregnancy for 3 to 10 years if you use the IUDs. And another one is the permanent type of contraceptive methods. These are the tubal ligation, a surgery that prevents a woman from getting pregnant, which is permanent. And vasectomy is a surgery that prevents a man from getting someone pregnant. It is permanent. So these are the permanent type of methods. The couples they have completed their families, they can use the permanent type of contraceptive methods. So they are free from the fear of the pregnancy, or we can say the unwanted pregnancy. And there are some of the natural type of contraceptive, or we can say the man-made your fertility awareness or body temperature awareness methods to avoid the days of the conception. And these include fertility awareness based methods. They are also called natural rhythm methods. They involve tracking the woman's fertility cycle and avoiding sex or using various methods on the days when she is most likely to get pregnant. This method have may have higher pregnancy rates other than other types. And the lactation ammonia method, a form of natural birth control for breastfeeding mothers. it relies on the new mother feeding her baby only breast milk for up to 6 months and having no periods or stopping during the time. And another one is the withdrawal. This is also the natural methods, and it is the time of intercourse. They can men can withdraw the sperm ejaculation inside the female reproductive tract. During intercourse, the penis is pulled out of the vagina before ejaculation. The goal is to keep sperm from entering the vagina, but the sperm can leak out before the penis is pulled out. So it has drawbacks or cases failure rates. So another method is after the intercourse it can be used called as a emergency contraception. So emergency contraception methods they use generally in the form of hormones, steroid hormones, or may work after 72 hours of unprotected intercourse. Emergency contraceptive pills reduce the chance of pregnancy after unprotected sexual intercourse. Common situation in which emergency contraceptive could be used include forgetting to take several birth control pills on in a row, having a condom break or sleep off, or not using a birth control method during sex. It also can be used after a woman has been raped. When are hormonal pills which the woman takes as soon as possible after unprotected intercourse. Emergency contraception is not a regular method. So they are used when they unprotected intercourse or they have to chance the fertilization and to prevent fertilization process. Emergency contraception is not a regular method for birth control, but it can be used to prevent pregnancy after unprotected intercourse or if a condom breaks. There are two types of emergency contraceptives. These are copper IUDs, which is a small T-shaped device that provider insert within 120 hours of unprotected intercourse. How does emergency contraception work? Using emergency contraception does not cause an abortion. An abortion ends in existing pregnancy. Emergency contraception prevents pregnancy before or after. So, in fact, emergency contraception usually prevent the fertilization process. Uh, Emergency contraceptive must be used soon after unprotected sexual intercourse to be effective. If they use within a time period, they are more effective, and that after the or delay the time period, they are not effective or they have some failure rate. Emergency contraceptive must be used soon after unprotected sexual intercourse to be effective. It does not work if pregnancy has already occurred. So they are not a abortifacient agent. If someone gets pregnant, then there are several types of hormones, injection, or other the natural products they can be used for the to expel the developing fetus from the uh, endometrium. So nowadays it is a practice to be a safe and long-acting contraceptive to be used. So emerge new techniques that is the immunocontraception. So what is the immunocontraception? As you know that it is birth antigen antibody reactions and the immune system of the body. Immunocontraception is a birth control method that uses the body's immune response to prevent pregnancy. So immunocontraception is the use of an animal's immune system to prevent it from fertilizing offspring. जैसे कि आप जानते हैं ये है कि इनको इम्यूनोकंट्रासेप्शन में ये सबसे फर्स्ट जो टारगेट होता है तो ये है कि टू प्रिवेंट fertilization. It means to prevent the contact of gametes. They can use to affect the gamete production or the and functions, etc. Contraceptives of this type are not currently approved for human use. Typically, immunocontraception. They are not approved for the human use because they have some type of side effects like irritations, allergies, been reported in the studies. Uh, however, they are reliable contraceptive agents. Typically, immunocontraception involves the administration of the a vaccine that induces an adaptive immune response, which causes an animal to become temporarily infertile. Contraceptive vaccines have been used in numerous settings for the control of wildlife. However, experts in the field believe that major innovations are required before immunocontraception can become a practical contraception for human beings. For the immunocontraception, two proteins or analogs have been developed, FSH and Zona prokind Zona palasoda proteins. They can be used as immunocontraceptive. In the field of immunocontraception, we can a long way during the last decade, Procaine, Zona, Pelasuda, PGRP, and GNRF vaccines have been highly successful in the controlling wildlife population, domestic pests, several species of zoo animals. The SCG vaccine has shown potential success in human clinical trials in humans. So why is the Human Society of the United States for sponsoring research in immunocontrol? As you know that United States invest a long, very um, they invest a huge of money for to develop immunocontraceptive methods. The Human Society of the United States believes that immunocontraception may offer a humane, non-lethal solution to conflicts between people and wildlife in urban and suburban areas, as well as solution of local problems of animal abundance. Immunocontraception can also help to reduce the overpopulation of captive animals also in zoos and other facilities in future might play a role in controlling dog and cat overpopulation. uh, what are the current objects of the Human Society of the United States Immunocontraceptive Program? And the HSUS is working to develop the Procaine Zona Palasoda immunocontraceptive vaccine into a safe, effective, and practical tool for the control of wildlife population. To make this goal a reality, HSUS partners are working including field techniques, demonstrating the effectiveness of immunocontraception in the field, refining the vaccine manufacturing process, and developing training standards. So these are the two or three vaccines have been developed and they are nowadays in clinical trials for the human use purpose. And what is the PGIP? PGIP, as you know, that proprine zona pellucida is a protein that occurs naturally in pig ovaries. The HUS and other are conducting research to develop synthetic long-acting formulation of PGIP. Zona Stat H is produced at the Science and Convention Center in Billings, Monitor. How does PGRP prevents pregnancy? Jonah pellucida proteins surrounded the unfertilized egg of the all mammals. The sperm must attach the sperm. It is the fact that the process of fertilization. And in the mammals or especially in humans, egg is surrounded by the protective or covering layers. So, Jonah pellucida, corona radiata. So, Jonah pellucida protein surround the unfertilized egg of our mammals. Sperms must attach to the sperm receptor site along the zona pellucida before the egg can be fertilized. Then pig zona pellucida at the PZP is injected into female animal. Her body produces antibodies to it. These antibodies attach to the sperm receptor sites where zona pellucida produces sperm from attaching the blocking fertilization. So the fertilization process can be blocked by the PGRT administration. So how long does PGRT last? कितने टाइम तक कितने पीरियड तक इफेक्टिव हो सकती है, ये इसके बारे में ये स्टडी की गई। For gonna start as two injections are given in the initial year, followed by animal boosters. However, one shot pediatric vaccines that last 2 or more years have been tested successfully on wild horses, deer, and other species. If Prochyne gonopellucida protein, the only immunocontraceptive being tested. Other immunocontracectives were developed by the other researchers. The most notable these in the Gona, which was developed by the USDA, APHIS, National Wildlife Research Center in Port of Collins. So nowadays, the new experiment is conducted on this PGIMS. Is PGIMS experimental with the achievement of EPA registration for PGIMS supported by long part of success on the captive animals. जो पीजर जो है वाइल्ड एनिमल्स है और दूसरे एनिमल्स हैं उन पर बहुत ज्यादा इफेक्ट देखी गई है। Then now start the for the human trial or for the use of immunocontraceptive for humans. So as you know that their first target is to prevent the production of gametes, or after that second target it may be the prevent the process or block the process of fertilization. In the history of immunocontraception started with the sperms as the first target. So sperm it is the most suitable and we can prevent the process of spermatogenesis or you can you can reduce the number of sperm production. Also helpful the, to achieve the contraception. It started 1899 when Nobel Prize recipient Karl Landsteiner from Austria and Sir Merislav from Russia, working at the Pasteur Institute, independently demonstrated the injection of sperm from hysterospecies can produce antibody response. So it is a long journey which is started 1899 for the development of new contraception. And injection of sperm from blast heterospecies can produce antibody response. Following this, there are reports published in 19, and the injection of the sperm can produce a period of sterility, infertility in the injected animals. In 1929, Morris J. Baskin, a Denver-based surgeon and the clinical director of the Denver Maternal Hygiene Committee, used human sperm to produce reversible sterilization in fertile women. So after that, it can be also hope that if someone has the infertility, so if they can be reproduce or we can change the they can be used as a reversible contraceptive. A US patent was issued for the spermatotoxic vaccine in 1937. Subsequently, there are few preliminary reports that use bulk sperms instead of human sperm to induce antibodies in women from contraception. Between 1950 and 1970s, sperm immunization studies regained impetus for fertility revolution. So now, after this, yeah, 1950 and 93, there again or regain this immunocontraception development. The recent Nobel Prize winner of Physiology Medicine in 2010, Dr. Robert Award, was also very interested in immunocontraception. When he started the new journey, Human Reproductive Update, this is the most prestigious journal of fertility. So, right. This is the mechanism. Here the picture you can see the mechanism that mechanism of action that can produce successful immunocontraception in male and females. You can see here the anti-sperm antibodies can induce and they can changes and production of antigen anti-sperm antibodies and neutralize the antibodies enters in the human. So male reproductive fact that they can achieve immunocontraception. They can the sperm can produce, but they are not ready to fertilize a single month. So, and there are several changes in the structure and functions of these sperms. And in females, the antibodies or antibodies hormone they can change the production of oocytes or they can change the uh, outer covering layer of the donor follicle. So, potential vaccine candidate it may be to prevent the production of ova or fertile ova. And they are they are tried in the pig, horse, and other wildlife. So this is a mechanism of the action of immunocontraception.
Hello. Yes, sir, we can hear
Hello.
you.
Yes, yes, yes. Oh, sorry. Yes, yes. The contraceptive vaccine led to generation of human or and cell-mediated immunity. As you know, the antibody and the injections depend on the and these vaccine can be designed to inhibit production of lipids that is and over and function of gamete that is gamete outcome that means pregnancy. So they can affect the production of gametes or functions of gametes. So if the production of gametes is reduced, and the time can change the production of sperms or ovum in the wild animal. So it can be to get, achieve the contraceptive. Certain desirable property of contraceptive vaccine make this approach to a potentially attractive option for family planning program. कुछ ऐसे कारण हैं जिससे इसको जो इम्यूनो जो कॉन्ट्रासेप्टिव है उसको ज्यादा लाइक किया जाता है इसके बहुत सारे इस तरह के हैं लेकिन ये जो कॉन्ट्रासेप्टिव वैक्सीन है ये जो है ये इकोनॉमिकली बहुत ज्यादा महंगा है. It is not easy to like just use of barrier method for male condom or female condom. It is a costly method. Economic easy usage, tolerance, reversibility, less failure, and freedom from mechanical devices or exogenous hormones make this approach an attractive option for family planning programs. Moreover, infrastructure for mass immunization development, most of the developing nations make this concept as exciting propositions. The sperms was the first target for new construction in 1899 when ये मैं आपको बता चुका हूँ. So during 1970, the scientists began investing various other targets besides sperm. सबसे पहले जैसे कि स्पर्म को टारगेट बनाया गया था और उसके अलावा स्पर्म के अलावा ये भी इस तरह की सबके बच्चे तरफ के ट्राई किए जो कि क्या अपना एक सेफ हो इकोनॉमिक हो और लॉन्ग एक्टिव हो लॉन्ग एंटर इनको और यूज किया जाता है. द वेरियस मॉलिक्यूल्स दैट आर बीइंग explored for the contraceptive vaccine development at the present time are summarized in three categories जो उनको जैसे गैमेट प्रोडक्शन गैमेट फंक्शंस और गैमेट के आउटपुट के ना. So vaccine targeting gamete production they can use in the form of GnRH, FSH, and LH. Gamete functions sperm antigen and oocyte donor cell. That is the JLT which is used for the gamete function interruptions. Gamete outcomes SCG the vaccines of SCG that can prevent the conception. As you know that SCG production is essential to maintain the early pregnancy. This success, their success in limitations and current status are vaccines targeting gamete production, namely GNRH vaccines and FSH, LH, and RH. They are synthetic analogs also. They affect sex steroid because they block LH FSH secretions The genotype vaccines have been developed by several pharmaceutical companies as substitute for castration of domestic cats honored by the rules. These vaccines can also be used in humans, especially for non-contraceptive purposes, such as decreasing androgen, prostatic hypertrophy, and carcinoma in men. So, immunocontractive methods can have some other purposes we can use. They can also be used in humans that require decreasing steroids, such as in uterine fibroids, polycystic ovaries, endometriosis, and precocious puberty. The vaccine target which involve functions such as antigen and Rx constitute the most promising and constitute contraceptive vaccine. So sperm is the most suitable target. To develop contraceptive vaccine, develop anti-sperm antibody process is the most reliable or suitable method for the contraceptive or in the this is the table. You can see here the different kind of the methods or the create the organs or the production of they are used for the immunocontraception development. GnRH used in female that causes impotency, FSH male and female causes in male causes oligospermia and female it can affect the production of ova and egg. LH male and female causes impotency, SC in female difficult to achieve, and they can prevent the or it can implantation, ZT feeling to the reversal options. So there is a limit of this immunocontraceptive. If they use immunocontraceptive methods, it may be possible they are a permanent type of they can affect the functions or the mechanism of the physiology of conception of so, what are the target and types of immune contraception? Contraceptive vaccine can be broadly classified into three categories. जैसे मैंने बताया आपको तीन कैटेगरी में हम इसको डिवाइड कर सकते हैं इसके अलावा कुछ ऐसे हार्मोन्स हैं प्रोटीन हैं जिनको हम contraceptive वैक्सीन के कैंडिडेट के रूप में यूज कर सकते हैं गैमेट फंक्शंस
You're now connected to Poststone 1000.
Sperm antibody. So, anti-sperm antibody mediated infertility provides a naturally occurring mode to detect low how affecting my birth hormone. Vaccine based on genetic sources are clear efficacy in producing potential effect, but make into two currents affecting sex space and they have been successful in anatomical population if we are listen of single human species. Plastic targeting carrying out from primary focus of the executive molecule as vaccine of the static million
Dipu, Dipu,
most effect for using whereas users and then purpose of increasing the feeling gains in about something being various to uh, because noticed that
ये स्पीकर नहीं काम
child
कर रहा है क्या?
we can do it can
ये
so
लैपटॉप
this
की
is
आवाज उस स्पीकर में आ सकती है, नहीं आ सकती
the
है?
model how can body
तो मैं अभी
you can see here there is from antibody from you can blood test is very concern and and it can be checked it can be checked after the
कनेक्ट तो हो गया,
care service and also the
स्पीकर तो
antibiotic
कनेक्टेड है.
option and hormone plus process. So this antigen anti-sperm antibodies can these antibodies for infertility and no antibody female antibody antibodies uh, can reduce sperm production, sperm transport, sperm mobility, and also do evaluate and the process of the activity can prevent or reduce fetal development in early stage. So the process it blocks the partition in the endometrium. Mechanism of action in culture is induced through connection vaccine. You can see in the picture where there is some character inside the egg and country pre-vitamin is in Europe and Pakistan. Antibiotics from Asian vessel fluid or intra antibody force antibody force to all creative response. Here this is the enter if they use anti-sperm antibody or and the like outer layer or outer egg the donor cannot be burned. It be prevented by the action of anti antibiotic discharge. So
Yes. Thank you, Dr. Pratap, for such a nice talk. It was very informative. Uh, Thank you so much.
Hello.
Yes. Hello, can you hear me? Hello. Can you hear me, Dr. Pratap? हेलो
हेलो
हाँ सर सुनाई दे रहा हूँ मैं हेलो 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 हेलो
हेलो
हाँ जी कैन यू हियर मी हेलो
आवाज आ रही है ना सर?
हाँ मुझे तो आ रही है आपको आ रही है
हाँ आइए अभी आइए
हाँ ठीक है हम आपका बहुत शुक्रिया अदा करना चाहते हैं इट वाज अ वेरी नाइस टॉक
थोड़ी कम आ रही है आपकी आवाज कम आ रही है
ठीक है सर एनीवे थैंक यू वेरी मच फॉर योर टॉक and we it was very informative and the students have really liked it we would like to move on with our function thank you very much I'll talk to you sir later bye connect हुआ भाई कोड डालूंगा hello I have sent okay okay good morning Dr. Devinder can you hear me
Cannot hear you.
you cannot hear me
Cannot hear you.
okay just a second hello hello hello
You're not audible.
hello hello hello
Yes, please.
Not audible at all?
And
Hello? 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 Can you hear me now? Hello? Hello?
Manju, I can't hear you.
Not even now? Can you hear me now? Hello?
That,
Can you hear me, sir?
Manju, I can't hear you.
Okay. क्या हो रहा है भाई? हाँ? क्या म्यूट हुआ पड़ा? अच्छा. Hello, can you hear me now?
Hello.
Yes, can you hear me?
Better.
Okay, so great.
Better.
Let
I
me
mean,
introduce.
if I am audible, then I think it will work.
Yeah, you're perfectly audible, and I think there was a technical glitch and it has been rectified. I hope you can hear me now. Can you hear me?
Yeah, little bit, yes. It's enough to understand what you're saying.
Okay, so Dr. Devinder, actually we can hear you perfectly. Maybe some error from communication from here to there. There is a error, but we can hear you perfectly. So I'll just introduce you to our students here. We have our own students from Pandit B.D. Sharma University of Health Sciences, and the speaker over here is Dr. Devinder Sehgal. He was a he has done his masters from Delhi University, followed by which he has done his MSc and PhD from Jawaharlal Nehru University, following which he had his postdoctoral studies in NIH, National Institute of Health, and after that he is back again at National Institute of Immunology. Let me tell you, National Institute of Immunology is the best institute working in the field of immunology, and Dr. Devinder Sehgal has held the torch for molecular immunology in this institute. So I expect a big round of applause from all you students. And over to Dr. Devinder for his talk. Welcome, sir.
Hello, Manju. Indeed, a pleasure to be talking to you and your colleagues and students. So, thank you for joining. And I would try and share the slides and then we go on.
Can you share your screen?
Could you please confirm whether you're able to see the slide?
Not yet.
Hello.
Not yet. Yes. Yes, we can see. Yes, we can see you. See your slides. Please go ahead.
Yeah, okay. On this occasion of the National Immunology Day, I welcome you all. As Dr. Masood mentioned, I am a scientist at the National Institute of Immunology, and I work in the area of molecular technology. So, what I'm doing in the next 40 minutes is to try to give a flavor of immunology, and I hope I to stand up to have a demonstration at the station. Better appreciation for the area of technology, and I gather that one of you are students, and you would be typically dealing with various institutions in the years to come, and I'm sure your interest and other structures will be talking to you about completely to how to work people. Okay. So what I would be discussing with you in this study that has been found really interesting. And we will I'll just be walking you through the system. Okay, now since the immune system is there to protect the disease, and this is from various incidents that occur. One of the main who is infectious diseases which come in multiple forms, and and also please remember that the immune system is not just to human beings. Malaria is activated to answer. So, for transmitting pattern to students, I've been talking primarily about human or human and just to give you some self stuff kind of about that kind of fun. One could have a situation where you have various fungal pathogens, viruses, bacteria, bacteria or just to make up with other aspects that are other kind of pathogens that may be involved. And all of these are trying to gain access to the system to expect the host or for propagation. And obviously, the human host is not is trying to uh, the human. So I hope the site the change in site is going up. Can you confirm? Uh, can Himanshu or anybody else can tell me that the consider immune system side on experience?
Well, we are only seeing your first slide, that is Immunology Day, April 29.
Also, I will just put my slide. So I'm going to change it back to the now this is the slide which has the title of the talk. And can you see this? Taking three from the title.
No, I just see the very first slide, that is April 29 Immunology Day.
Okay, I'm going to try and send you connect 1 second because the slides are not moving.
Yeah. In the previous talk, I had sent the link and it worked perfectly fine. Okay, it is visible now.
So,
Yeah.
it is visible?
Yeah, perfect, perfect.
Okay, so we can go to the next slide.
Yeah, sure.
Another slide. immune system and all that.
Now I'm seeing your title. Yeah, it is working. Good. Please carry on.
So this is can you see this? I mean, I'm just describing the side
Yeah,
immune system. Then we have something called host. This is visible.
yeah.
Okay, all right. So sorry about that. Essentially, what I was describing is that the even host immune system is the target of a lot of pathogens, and the immune system within the host tries to defend the host from these different pathogens. Okay, and it's a very complex and a huge universe out there, which is causing which has to be dealt with. And I will not be going into the details how the deals with this. The recent example of SARS-CoV-2, which causes COVID-19, all of us have experienced. Not only is it a very severe pathogen, but it is also has it's an ever evolving species. And remember, so it's a moving target. We have various kinds of strains. Delta variant, for example, caused a lot of havoc within the country. Half a billion people have been infected with this virus over the years, and millions, 6 million odd people have died. And obviously, we understand that these numbers may be more than what they are. So the pathogens can have a lot of diversity. They are ever evolving, and so it's kind of a moving target. And also to keep in mind that uh, they could be, you can make drugs against it, and then, they can develop multi-drug resistance, or you can make vaccines which may become less effective. You have to, come up with new. So this is a ever evolving moving target, and this is not just restricted to SARS-CoV-2. Many of the pathogens which now may not have caused as much havoc in such a short time may have all of these properties. Okay, so if understanding the immune system becomes extremely important. So for the purposes, you consider that we are medical students. I have picked up a disease which is particularly relevant to India as well as today's presentation. This is a bacterial pathogen called Streptococcus pneumoniae. For colloquially, it's called pneumococcus. So I would just give a little bit background before we get into a little bit of the story as we go along. So this is a pathogen which is airborne. It spreads much like SARS-CoV-2, and the bacteria, once a healthy individual inhales, um, uh, particles which are infected with bacteria, the bacteria colonizes in the nasal pharynx, and this colonization can be, basically the pathogen can be there as a symbiont for ages, and without any showing any symptoms. So it is basically colonize the nasal pharynx, and most of us actually would be carrying the pathogen. But that it's innocuous as long as we are healthy. But under circumstances where circumstances where the host is compromised because of let's say another infection with virus and the immunity is down, the pathogen from the nasopharynx can gain access to other parts of the body which are generally sterile. Okay, I mean these include, for example, if it gains access to the blood, and it causes sepsis, bacteremia, or you could also find its way into the lungs, causing pneumonia, and subsequently down the lane it can also infect meninges, causing meningitis. So there is a spectrum of diseases that are associated with it, and the burden of and the high risk groups that are in this case are infants who are less than 2 years of age. In terms of number of deaths in this age group in the world, India has the distinction of we will have a little bit of discussion on the pneumonia as we go along. So just this is just to translate that we are now we are very students just to give a sense of how things happen. And this is just a cartoon trying to show the interaction between the pneumonia and the epithelial cells that are present in the nasal grains. And down below we have the epithelial cells, and epithelial cells in this area would have a nucleus layer, and there are proteins or glycoproteins present on the back on the epithelial cells, which you know serve as receptors for proteins present on the back pneumococus. Okay, and these interactions are important for the initial adhesion. As I said, the, the pathogen colonizes in the reservoirs. Okay, so there are a variety of interactions that happen, and these are very important for the biology, and then subsequent, the pathogenesis caused by the by the bacteria. Okay, so and as you would notice that these various proteins, the PSA, for example, CBA, these are examples which have been well documented in the literature as being contributing to the overall adhesion with epithelial cells. So there are molecules present on the surface which may be of interest, and in our own lab, we are looking at a variety of what are called virulence factors that may present on the surface of the bacteria. Uh, I think here are some of the summary of particular different molecules, which may be mostly protein, but also polysaccharide, which is the principal virulence factor that forms a structure around that. Okay. So I will not go into the details of it, but just to tell you that they have variety of virulence factors that can be contributed by variety of things. For example, they can be crossing. For example, we have in the lower part of the picture, the immunology, which is mixed, released by the pathogen, goes into the host cells and bites the host cells. We also have molecules which help the adhesion, and as you would go along, there are other. So our lab has been looking at some of these proteins on the surface. Uh, what I will be describing to you is some effort to look at what is released when the protein surface conforms to you also have components that will be released by the bacteria, which could also be contributing to the process. So one of our students in Chile was interested in looking at what is being released by the okay. Now just a couple of terms that might be useful for understanding what we've been describing in the so pneumococcal is an extracellular bacteria. Okay. And what it does, by virtue of being extracellular bacteria, the main form of protective immunity is typically antibody mediated, which is the humoral arm of the immune system. And the way the pathogen is cleared is by a process or mechanism called opsonal phagocytosis. Okay. Uh, One of the major contributors to this process is our antibodies. The way it typically works is, I'm sure you have heard about phagocytes. In the context of the immune system, you would have, for the examples, include macrophages, neutrophils, I'm sure you are aware. Here are the major cell types present in the blood. So they have on the surface of both the neutrophils and macrophages have what are called capsid receptors, which are the stem part of the Y-shaped antibody molecules that we are all aware of. And these antibodies, if they are directed against the surface of the neutrophil, then they pick it up and then phagocytosis, which is the chain of kind of generic to mechanical cells, and within the cell they are okay. I describe a little bit of this. So this process is extremely important for as the far as the host is concerned. And please remember that the interaction with the host and pathogen is basically a war and an ultimate host is trying to get rid of the pathogen, and on the other side, the pathogen is trying to evade the host system or come up with strategies to avoid the uh, the immune attack. Okay, so these are working in opposite direction. Okay, so this is just a simple cartoon describing what happens when a neutrophil comes in contact with the microbe. Okay, so as I was saying, if you have molecules of the microbe which against which antibodies are there in the host, the active molecule in turn can bind to specific receptors on the surface of the bit of good learning, but these are then internalized into bacteria, and then we have the of like particle and then metabolize them, and the can not go repeated at some point time within these carbohydrates when you have the variety of mechanisms which should lead to the peak of the internalized microbe within the process. Okay. So this is the mechanism that is used for a structural bacteria with target. So just overview of what my other is looking at. We are going to questions since we have been looking at the positive responses and we have very looking for the pathogen evasion strategies that are there. We have interested in facts of surface proteins also because when the pathogen comes in contact with the host cell, the pathogen alters the cellular and multi processes in the whole cell and part of the perspective at novel factors.
Excuse me.
So,
Excuse me, DS.
Instead of giving a lot of data, what the program will be doing is that walking
Excuse me.
of journey of how it panned out in the lead investigating in this that is this journey, and this is to help students understand that the ups and downs and also excitement and then situations where you say I don't know what to do next. Okay, so this is just a description for this, and if there is something which is pressing and the student would or student or audience would like to answer, please feel free. I will be happy to answer. Okay, so as the story started with the idea that we are several of us, several of the other students have been looking at what virulence factors that can be present on the surface. And so she came up with the notion that why don't we look at the secretome, the component of components that have been released by the pneumococci into into the medium, for instance. Okay, the process was reasonably straightforward. What she did was that she cultured pneumococci and piloted down the cells, looked at that was there, concentrated, and that's what we called the secretome. And again, there were a lot of other experiments that were done, on to understand how complex this secretome was, and she did a lot of two-dimensional cellular performances. There were 150 to 200 odd protein banks that we could find out, and uh, uh, then she also looked at uh, when she used this secretome again, she found that it seems to be degrading DNA. Okay, so if you take DNA and incubate it with and the DNA was getting digested, and which was not what one would expect. The DNA DNAs are there, which are a part and parcel of the regular propagation and within the host cell. There is DNA replication that is happening, and as part of the various repair mechanisms that are there, you would see that. Okay, so this was one a surprising finding to begin with, and so what she ended up doing was that she concern here was that perhaps maybe the bacteria, once it grows within culture, maybe there is a small fraction of cells that may have actually lice, and what we are seeing is DNA activity from the lice cells. So she did experiments where she took bacteria that were in early log phase. That's the time when the cells are dividing and healthy and they are in great physical shape. And we did continue to see DNA activity, which basically will show up on the gel as a clear lane. You have a DNA band which after if it is digested, it is broken down into smaller pieces and then it basically runs out of the gel. Okay, so in another set of experiments, she actually deleted a gene called autolysin, which is supposed to be, causing cells to lyse at during the stationary phase uh, of this growth. As I said previously, we have been looking at the early log phase for this experiment, and to her surprise, despite the fact that genetically modify the pathogen so that it cannot lyse anymore. Still, she was able to see. So that kind of gave us confidence that okay, the bacteria is being released by the DNA's activity is being released by the pathogen. Okay, so then again, if you go back to your biochemistry, you will realize that typically nucleases or DNAs have a requirement for divalent cations. Again, I will not walk you through the data, but just to tell you that if you get rid of divalent cations, the activity is reduced to some extent. Okay, so and if you since the DNA is also an enzyme, if you boil the cyclochrome and do the activity, that will get on. So all of that was telling us that indeed, this is some kind of a protein which has DNA. Okay, and if you do not kind of digest the DNA with the protease, the DNAs, you whose activity also thinking that it is protein fishes. Mixture. So now we have a mixture with us which has about 150 to 200 protein. Uh, how are you going to identify which of these next two proteins is the thing? Okay, so these were experiments which I remember the amounts that we looked at are at a very very tiny amount and was needed most significant medicine. So now this is such an experiment because I'll just walk you through a little bit so that it gives you a understanding of what really how it is done. So as I said, the 60 was generated and two stage polyacrylamide gels were generated. In one case, the DNA was already included as a part of the gel system, and the secretome was loaded, and you do electrophoresis. Okay? And the various protein components are going to get separated, and then these remember the DNA, as I said, was a protein as the said in the previous slide, and they will all get denatured. So when we run the gel, then we need to get rid of this component, the detergent SDS, that is present in the stage for one to be able to kind of attempt to nature, re-nature these proteins, and hopefully they will get back to activity. Okay, this is how the cell looks like. On the left hand panel, as I said, when there is DNA activity, you will see that it's clearing on the gel. And on the right hand side is a gel, if you get the pointer, is a gel which is without the DNA, and these bands correspond to these, but then kind of excised and then sent for what is called mass spectrometry based analysis. And so from there, since this is a one-dimensional gel, we are able to each of these bands that we sent for sequencing had multiple proteins, and then we had to go back and look at the sequences that we got that use bioinformatic tools and understand which of these potentially can initiate. Okay, so based on all of these analysis, we found two different proteins. One is called CAND, the other one is a nucleus protein. Okay, this is based on information that we could gather using bioinformatic tools that doing. So I think we then were trying to figure out this protein that is typically not considered for any. So how do there are obviously that is indeed as various activities. So what we found was that all in biology transcripts for a protein which is synthesized it has a enzyme that will help it to produce the nutrient and release it. This is very it carbohydrate, which is responsible for targeting the biggest partners or being treated out of the cell. Now, what we found was that there was a situation will be how if we do not have how to see how to at this point in the was conducted. How it would happen. So we went back to the about what would be the disease sector. So that was in the protein, none of these would have because it's for comparison of this 1/2 program to get the amino acid and typically in that D protein comes that and this is not the homology pretty much on the lower end. And so I think that was mystery number one. We got what we so we said on next steps, purify this protein that is the one that is there for this gene in the mitochondrial genome. So this was expressed and defined from E. coli, and so for those of you who are familiar with this, there are situations where when you are purifying, expressing a protein in overexpressing a protein in E. coli, they go into what are called bodies. This was one such protein. They just drops out of the cytoplasm into what are called expression bodies, and so typically proteins that are they lose their biological activity. So we we raise antibodies against this protein, and then we just wanted to make sure that this is whether it is present on the surface or not. These are some experiments that we did by using a method called flow cytometry. Again, I would not be going into the details for your per se. But what we did find was that this protein is actually not present. And if one were to simply genetically delete this gene from the bacteria, even those strains also don't only active or not so many presence in the surface. Uh, so essentially, what we are finding is that the protein is absent on the surface, and but it is present in the supernatant, and this is not because of crisis. We first step was that while we were figuring it out how does it come out of the bacteria, uh, how is it released, when it comes to biochemically characterize this to look for nuclear activity as different as you would think, as was originally mentioned. So this nuclear activity one should be able to demonstrate that the DNA is getting fragmented in its presence. Okay. So we had an assay in place, and as I had mentioned previously, we also have various we looked at various cations, whether they have any differential activity, they augment activity in the big way. So again, if one were to go back and get the panel on the right hand part side of the slide, so what we basically find is that one we get the calcium cobalt variety of solvents, and what we found that there is some missing basal inherent activity present with the nucleus acting ever at times increases with a little bit, but not substantially. However, we see some which were decreasing that. So, but one of the issues, one of the other aspects that has to do is the connectivity. Also, we were able to demonstrate that it has that activity, but emails can be of all variety. Okay, some of them are called nucleases, some of them are endonucleases. So, in different kinds of DNA substrates that provided for and we were trying to find out whether how it okay, or so what we saw was that in two point which is next successful, then there are no cuts or leaks and we put in digest, which basically means that if they are circle, if it connects, that means it will actually connect. Okay, but so what I've told you so far is good routine, just 1788, also the factors that we has some intrinsic basal level of the activity. This activity is there in direct content. And if you add and this protein is an endo deoxyribonucleic, which means that it can digest gaming, nothing else. So while these studies were being done in looking at literature of other species, found that there is the early initial reports which suggest that bacteria is able of budding out very small vesicles from their surface, and these small vesicles also shown up here. They are nanometer scale vesicles. So essentially, if you go back and look at cartoon, you will bud out much like the way of bud out, and these are in the order of nanometers. The diameter is in the order of nanometers. And when they are budding out, they can actually carry components as part within. So in this illustration, what we are seeing is you put enzymes, you put DNA, you put oxides, a variety of things, to get they get packaged into this. So for most practical purposes, it would kind of look like virus. So these are the two extracellular vesicles because they are being, you know, released by the bacteria. Okay, so how do you, so she thought, okay, no, I mean, since we are not figuring out how this protein is getting, you know, sent out of the cell, she went back and then did what is called differential centrifugation. So you throw the bacteria, take out the supernatant because it would be supposedly released there, and these are as extremely small vesicles. So you have to do, high speed configuration, and then, do various kinds of gradients, and in certain fractions you will get this particular EV, extracellular vesicular fraction. Okay, so she then isolated the fraction, and then she looked for that whether this protein was present with the EV fraction, and how you do that? So she used the antibodies that she had placed against the protein, and indeed she was able to find this protein there. So which kind of positive release, and also very surprising that while the bacteria is using a vesicle to cargo or transport the nucleus out of the cell. Okay, again, what we then went on to do was that a mutant strain was generated which lacked this good protein, the 1788 protein, and extracellular vesicles were generated from the what is referred to as the wild type strain, and then also from this mutant which lacks this particular protein, and the presence of this protein from the EV fraction from all these cases was looked at. And in terms of enzyme activity, what was seen is that there was hardly any activity in the case of the EVs from the mutant because the lacks protein within there. So which kind of said us, they kind of give us a bit more confidence that in the absence of the thing which acts the 1788 gene is unable, it does not produce DNA activity. So again, the sense of it is now think about this. We have a bacteria which is releasing a small vesicle. That vesicle has this protein 1788 and the enzyme activity. Okay, so what is the physiological function of protein? I mean, if it is doing it, that's okay, but why would any bacteria do this in first? Okay, so at this point in time, Hina was wondering what to do. I mean, we are seeing this, there is experimental evidence pretty convincing, and but why would any bacteria do this first? Okay, and again, just for the students, uh, uh, these are activities that are happening elsewhere in other fields, and one of the studies that I came across was a process which was described relatively recently called necrosis. This is NET stands for neutrophil extracellular traps. I'll just describe them in a little bit. So what we have is that again, as medical students, you would know that neutrophils have a by kind of probe nucleus. There are a lot of granules which are flooded with various antimicrobial peptides, and in this when these cells are in this uh, in these cells are uh, stimulated, they release these granules, and alongside they also release DNA chromatin out of it. And these components, which include variety of peroxydases, elastinase, and other antimicrobial peptides like SL37, they trap the bacteria within them, and because of these antimicrobial activities, they are then. Okay, so this is the process called necrosis. This in addition to the phagocytic activity that I was mentioning. Okay, so again, I will not walk you through the protocols, but essentially what was done is that we were taken and they were stimulated what is called DNA, and they would release this nets, and so the nets that these are experiments that are done on the slides and here.
You plugged into Nirvana. You're now connected to Boatstone 1000.
So that's what the same thing. So we also deleted that thing as well, and we found that the contributes as little bit with activity, but it's not very significant there. Okay. Coming back to the what implication does it have for the pathogenesis? So we did some experiments with the mouse and the model of our studies, and this acids that are shown here. We injected mice with the wild type and also fiction. Getting this and we are looking at the bacterial inputs in various organs, lungs, bladder, and what we find is that in the absence of this gene, the bacterial loads uh, come down. They are regulated to handle some smoother. When you go back and look at histology of the lungs, I remember called as one of the pathologists, and what we found was that pathology in the mutant is lesser than what we find with the wild type. And again, we did some experiments which were to check the virulence properties of this bacteria, and what we found was that the post in the absence of the of the gene, the ability of the substance to cause disease is compromised. Okay. So essentially, the and conclusion that we do from this experiment were that SPDM score 1788 efficient strain was attenuated in virulence and had lower bacterial loads in lungs and blood and skin in urine sepsis model compared to the wild type. Okay. So just to summarize what we found was that the difficulty that we were facing is that how does the nucleus come out of the whole cell. And as I described previously, that they are packaged into these extracellular vesicles and they're transported out of the host in this EVs. Okay? And data from another lab said that there is a nucleus on the surface. It is called PGIMS. So the working model that we had was that once the neutrophils are activated and they release this nets, Limnococci uses two strategies. One is that if it is in physical contact with these nets, it uses this NDA nucleus to digest it. And the other strategy was that it releases an extracellular vesicles which can act at a distance and bearing this nucleus, and these nucleus can digest the nets and then help immunococci escape from nets, because this once it is stuck there, it's a very lethal form. And for immunococci is concerned, there are a lot of different enzymes and different antimicrobial mechanisms, including peptides that are going to digest this, which is essentially kill immunococci. So,
excuse me. Excuse
so
me,
in
DS.
some sense, coming back to the title of today's presentation, derives from this aspect that the neutrophil is kind of holding the bacteria in a way which is going to kill it, but what pneumococci is doing is that it is coming up with a mechanism to digest the DNA part of this chromatin and release itself from there, and then escape and infect other parts of the body. Okay, so the carry-home message here is that 1788, which is also referred to as CAT, is the novel extracellular vesicle associated endodeoxyribonuclease that facilitates escape from nuclear extracellular tract and is essential for fulfillment. Okay, so this work was primarily spearheaded by one of the former students in Athens with help from Immunology. This was funded by the NII Intramural program, and thank you so much. I will be happy to take it.
Thank you, DS, for a very nice and elaborate talk. I hope I am audible to you. Hello. कहाँ है? कर भाई.
Hi.
Hi, can you hear me? Hello.
Yeah. Yes.
Yeah.
Go ahead.
So, thank you very much for such a nice and elaborate talk. Basically, I had initially requested you for a very down-to-earth talk, but yeah, molecular mechanisms were very much highlighted because our students they don't have this much exposure even to a gel electrophoresis. So, getting into all these stuff is really too much for them. Okay, so one very simple basic question for you as well as for a clinician who are here. Um, Basically, our immune system is like an army. So how much can we trust our army? Do you get my question?
Yeah. So yeah, you're right. It's one can visualize immune system as an army with lot of as components and arms, and so it is designed to deal with lot of different kinds of assaults, as I was mentioning. That you could certainly have situations which can which will overwhelm the capabilities of the immune system. Okay, and it could as it's like saying that you have a much bigger, I mean, typical example that we saw. I was the COVID-19 story, where you were hit by something which you cannot even figure out. Your body is not prepared to deal with it, and that's one. Uh, but what we don't realize is that one is under the host or the immune system is under threat all the time. Okay, the best comparison would be to look at a situation where a person is immunocompromised. Okay, or an animal model for that matter. Let's say we are seeing a bad deficiency mice strain. They are susceptible to mutations of susceptible to infections of what you would not even consider as infection. You would find them all the time. Okay, let's say somebody cannot make antibodies against any antibodies at all. Those people would get infection every second day. Okay, so the immune system is doing an amazing job. Uh, uh, there are of course limits to what can happen. And again, these are all multifactorial, meaning that your nutritional status can also make a difference in your pathogen. If immunological thing, you could have a genetic deficiencies that can cause complications. So there are limits, but I believe, considering that we have immune system per se, humans have survived for such a long time. If multiple things have contributed, immune system certainly is one major component. I hope I was able to dwell on some aspects.
Yeah, sure, very much. But the only thing is, again, with COVID-19 situation, one question always hindered my mind: how much can we trust our immune system? Suppose if we keep on over trusting them, and if at the end of the day one is no more, there's no point in having a trust on it. So basically, you have to estimate your health. Assessment should be there, uh, very frequent, in case of emergencies, or whenever there is a challenge uh, by a microbe. This is what I would take. Anyway, thank you very much. Very nice presentation. And
If there are questions in the audience, ma'am, you will have to bring it up, because I'm not able to hear very well. So the questions have to be related to me, or if there are any other points, comments.
Yeah, sure. Thank you, DS. With this, from bottom of my heart, and on behalf of PGIMS ROHTAK, we thank you very much for your excellent presentation. Thank you.
Okay.
Chalo, with this, thank you all for your patiently hearing. It was almost 2 hours you have spent here. I'm sorry about the technology that we have our disposal. ऐसी टेक्नोलॉजी से तो ह्यूमन जीनोम प्रोजेक्ट कभी नहीं हो सकता but I can't talk to him, but if you have some curiosity, then I would like to sorry, I can proceed. Okay, then that's इससे पहले कि कोई और निकल जाए, because the audience is very limited, then we have a group photograph of everyone. Please, everyone, please come on stage. Everyone, please walk on. जो लंबे लंबे है ना वो पीछे चले जाए, छोटे छोटे आगे आ जाए। रोज चल बैठते हैं, आगे वाले पीछे। एक मिनट एक मिनट। सोचा उसमें माउस है मेरा। हाँ? ना वो ठीक है, अभी मैं इसमें हट रहा हूँ बस।
at least you believe that you will sick. You never know what happened. But I think so that now this right now the fourth wave of COVID is going on. But have you seen so many deaths? Why? Because there was a very large scale vaccination in India. Of course, in other parts of the world, but India, they made the record of vaccinating maximum number of people. So your population is so large, you should record it automatically. Because otherwise countries are they have any problems now 50 crore, 20 crore, 25 crore, and it goes on. India has 140 crore population more than us. Now you may be aware that now India has become number one in population. China is also like the 25, right? So when you vaccinate so many people, automatically because of this, right? So I mean to say that so that your immune system keeps on boosting and works better, so you got the boosted immunity. You got the final booster dose. So after that, you fully believe that that you will not need any right? So I mean to say that this immune system is very, very important because it keeps invaders away. Invaders mean all the foreign organisms, microorganisms that body recognizes foreign. And foreign means that is not the part of the body. So it keeps on feeling that you know that every immune system, every immune cell, it has three elements. One is memory, second is recognition, and third one is presence. Because each one of the cell memorizes what happened. Because if it is invaded by some microorganism this time, so when it comes next time, it already knows that it invaded here. Or if it is new, it also knows it is new. So it has a memory. Second is the recognition. It recognizes. It recognizes. It has a mechanism to recognize any foreign element. And it has as well specification. It knows which particular organism is invaded. Right? So these three elements are very important, and these are the key pillars of the immune system. So when any one of these goes weak, that means something is going to be bad, and your system is going to be weaker. Because otherwise, also you know that if you have a friend and if you don't recognize him or her, then how will you feel or how your friend feel? You say, "ये तो मेरा बहुत अच्छा दोस्त था, बहुत अच्छा friend है, like that." There are implications to that. And especially, you know, like you know which particular friend, you react like that. You know which react like that. Right? So you just need that. So these three elements are the key pillars of the immune system, and they have to be strengthened. They have to be boosted. So you take different kind of natural aspects, or you take some synthetic supplements in your diet, so that you keep on boosting your immune system. So I mean to say immune system is very, very important. And as Dr. Mansu told and asked the question that how long we should learn from our army, right? Because these are the army of your body. But now the army structure is going to change. Now the Jawans are going to fight only for 4 years, right? The, some of ourselves are going to be active. But we want to send the permanent army that will fight for us, right? So I mean to say that you have to keep boosting your immune system. So for that you need to keep doing exercise. You need to find out the very positive person that because all these studies only useful as long as your body is supporting. Again, your body is not supporting you, you don't feel anything good. You don't take any interest in learning about anything unless you are yourself healthy. अभी आप जब तक बैठे हैं जब आप healthy तो मतलब तो विश्वास नहीं अभी तक interest छोड़ के आ रहा था कोई नहीं मिला था माइंड नहीं दे रहा था क्योंकि हमारा सिस्टम जो है ट्यूब है right? So it wants to be healthy all the time because our body is made that way. You want to remain healthy, but something happens, you know, sometimes something happens, you are invaded by some microorganism or you get some, you take some you had an accident, you never know what happens. But we have to deal with life most effectively by the drugs, by the natural ways like exercise, meditation, whatever. And then we get people. So ultimately, we are trying to do the immune system. And now Dr. Hemant is going to talk about antibody antigen interaction and how this plays important role in immunodiagnosis. What is immunodiagnosis? There are different ways of diagnosing disease. So one way of that is by using antigen antibody interaction. Right? Because mostly what happens in immunology, you study about the antigen response. Something is happening between the antibody, antigen, some interplay between antigen. So what happens when you do the diagnostic test? So that interaction between antibody and antigen is recorded, right? And we get some kind of marker, like if there is color change or if there is increase in the level of certain antigen or antibody. So we say that this is how we detect and the more right now with the marker for this, and he has a lot of experience with immunology and inherent sequencing technology. So he can imagine you how you can learn better what like thank you very much, Dr. Dhara. आप लोग ना एक ही जगह आ जाओ तो मुझे थोड़े दिक्कत कम होगी तो इधर उधर सब जगह देखना पड़ता है आंखें बहुत थक जाती हैं
Okay, so this is a very favorite slide of mine, and I discovered it just 2 days back. ऐसा कुछ भी होता है. What you see in these glowing things, bioluminescence. This is a phenomenon of bioluminescence, and actually these are planktons. Okay, and in planktons these are called as. Anybody knows the name of this plankton? The loud fractan is the name of this plankton. Do you know this? Sunan? So yeah, so why should we look at it? We should look at it and we will feel the need of this when I start my actual talk. And the talk is very simple. I'm just going to talk about ELISA. When we have this kind of sophisticated days, like immunology celebration or some WHO celebration, there are many big institutes that come and they deliver those might be exceptionally good people doing exceptionally well with good research, but then our audience is very different. If these are MBBS students, BBS students, physiotherapy or whatever students these are not very much into research. Still, you people are learning and you have some experience or exposure to some of the techniques. Basically, and those are very superficial. So, and we get even spoke about deletions and how they break the number and how there are those were hardcore immunology. I did ask him. He said, super simple talk because this I see this every year basis because our students just go back and we got very sophisticated because we started in 2016. That was the first time, and the speaker was Dr. Ray Chaturvedi. He was the physician who was president. He was the rheumatologist. So yeah, we had a lot of friends. That was a different issue, but. What about the audience? The audience was very layman audience we have, and we will always be having layman audience until and unless we are doing research in this institute. And as of today, we are not doing research. We are trying to get on to that platform of doing research, and we will be starting very slowly, and it will be a slow and steady ride. Yeah, we have some sophisticated machinery and equipment with us, but that is very much limited. And, students don't get to uh, know about it, or they don't know it. You don't know what is kind of equipment that next generation frequency is, because neither you have come to us nor we have gone to you. And two people, me and Dr. Deepak, work on this machine, and it is super sophisticated. We spent 3 days working tirelessly to get some results out of it. And we start on Monday by then, and then bioinformatics takes another couple of days. So by Saturday we are done with it, and we are absolutely exhausted. So that is the status of understanding these toxins. So I made it the simplest and simplest of it. But if somebody already knows ELISA, because ELISA is not a big thing now, many students know it. At the end, wait till the end, because in the end, you will be surprised to see few other techniques which I'm sure you would have not heard about. So yes, this bioluminescence. I'll come back to this bioluminescence in a little bit. So let me start the topic. ELISA test. Is everyone familiar with ELISA? सबको process पता है? So I will not waste much time. And actually, I had a what you call a quiz program to be started. If we had started at right on dot 9 a.m., then there was a quiz that was planned. But still, I was feeling it was too much for our students. So we, but because of time, we lost that program. But still, this was one question I could have asked: what does ELISA stand for? And then there is a further round of where the team would say, and I think everyone knows. So ELISA is nothing but ELISA enzyme linked. And so, what is the history? What, how was ELISA developed? और उससे पहले क्या करते थे लोग? हमेशा कुछ ना कुछ करते थे. जैसे PCR is very famous. PCR came into existence in 1985. DNA amplification लोग करते थे. And but the method was different. So here, before 1970, uh, radioimmunoassay was used, and radioimmunoassay it is called because there is a use of radioactivity, and so there was lot of radioactivity was used, and once the radioactivity is there, radiations are there. Not everyone is interested. Some people will just leave the field just because there is radiation, and some people, I mean, it becomes a little bit of choice. So, especially pregnant women will be kept away because they are those issues with the fetus. So, so there are certain who can handle it, who cannot handle it. So it was not liked by everybody, but yeah, those were very sophisticated instruments and everything. And then there was a reporter signal that was indicated, and then the signal was captured on a photographic film. So again, you have to have a dark room, and you have to way of capturing that thing. So very cumbersome. So in 1971, these people came up with the first radio immunoassay. And this is something, how much is the affinity with one antibody, with the two, what is the binding site suggest if there are multiple antibodies, there will be more of binding, and the binding will be very strong, that will be counted as affinity, and affinity can increase by uh, antibodies like IgM, because those are pentameric in structure or high in number. Now, these are the types of ELISA. I hope you might be knowing. Direct ELISA is very simple. The antigen is there, and the primary antibody that is coated that reacts with this antigen, which is very this antigen antibody interaction is very specific. It is just like lock and key. And once sequences, there is nothing that can break this binding. You throw the plate from top of a building down, still this bond is going to be staying there. And then this antibody is linked to ELISA, and then enzyme can react with substrate. So this is the simplest form of ELISA. And then people have harnessed this technology uh, for a, they have introduced a secondary antibodies like the primary antibody is patient sera most of the time, and now human IgG can be used as a antigen in some other animal like horse, rabbit, dog, cat, whatever it is. So, and the antibody that is raised in response to the human antibody, where the antibody is acted as antigen, will give rise to a secondary antibody. And this is a very good business. Like you need only one sheep, cow, lamb, dog, donkey, whatever it is, and you can raise lot of amount of antibodies, and these are secreted in the what you call serum of the animal, and these are really very expensive. So if somebody really wants to harness this technology, this is one simplest business one can have. Uh, Make a secondary antibody and then conjugate it to an enzyme. So this can be done. These are very simple biochemical processes. And now here you have an indirect uh, assay. And now there is something also called as the capture sandwich assay, where you have an antibody first and then you capture the antigen. There are certain antigens which you might be looking at, like cytokines in the blood. Those are not antibodies, those are actually protein material. So those can be captured by this capture antibody, and then there you get have a specific antibody to this antigen that will bind in turn, and then uh, just like the previous one, we will have a conjugated antibody, this is termed as, and then substrate with them. So this is a type of disease which people have uh, used, and it is very nice that nowadays students simply say we know ELISA, we don't want to hear much about it. So it's a qualitative technique and as well as a quantitative technique. How you want to use it, you have to devise your assays accordingly. I'm not going into this FAB segment sites. So these are the different types of antibodies. Everything is very clear. What are what about the reagents? So you would need a few reagents. You would need a polystyrene microtiter plate. What is a polystyrene microtiter plate? I'll show you in a video which I have after this. Then there are certain buffers because all the biological reactions have to provide a specific buffering reaction, buffering environment to the system. And then washing is very essential. Since there are multiple steps, you coat whatever you coat your microtiter plate with, you have to at the end wash it, which is very essential. If the washing is poor, you will see there is too much of background, and then substrate and enzyme, all these are needed. These are the samples which can be used for ELISA. This is very straightforward. Equipment, you will need pipettes, tapes. Uh, these are the equipment. This is a plate reader. Uh, this is an incubator and multi-channel pipette. All these things are required to perform. And this is a how does a ELISA reader work? This is nothing but a spectrophotometer, but a little more sophisticated. Where all the wells can be read in one go. And then this is different types of ELISAs again, pictorial review. Uh, these are the conjugated antibodies that are used. The conjugates are most 90% of the time you would end up using HRC or peroxidase, sometimes alkaline phosphatase. These two make up 95% or more of the conjugates, and then there are certain other for some other purposes. And then we have listened to the enzymes, and these are the substrate, and then whatever chromogen, and then there is a stopping end up stopping it. These are the different wavelengths. Now, since you don't see the color here, there's point just to talk about it. And finally, the best thing you can do is develop a kit. Once you identify a specific antigen, uh, make your own kit. If you are successful making your kit, then you are doing a big business in biotechnology. So components, all these things is understood. This is what a microtiter plate looks like. Basically, it is 8 by 12 wells. These are different wells in this small structure, and these are come to. So these are 96 in number, 8 by 12, 8 on vertically and 12, so 8 into 12 gives you 96. So if somebody is doing too many samples, then there is a 484 wells plate that is called as high throughput, uh, what you call ELISA. So this is what a microtiter plate looks like. This is the end of the ELISA, and you have positive, very high positive samples here, very high positive samples here. These are the light blue ones are kind of very low optical density, and then there are several of these which have no color at all. So these are negative samples. Okay. So this is a ELISA washer. Now, what are the advancements I'm going to say? So in this is a nitrocellulose paper, nothing but a nitrocellulose paper you have here, and this nitrocellulose paper is coated with certain vector antibody. So if you suspect a cell type that is releasing a specific cytokine, suppose IL-1, take for example, interleukin 1, you want to study how many cells are going to be secreting IL-1. So you will put an anti-IL-1 antibody on this paper, and then uh, allow to incubate for some time, and then uh, so the antigen antibody interaction, the cells will be captured, and then you can further process this just like ELISA, and you will see wherever your cells were bound, you will see the color changing there. So this assay or this test is done as LE spot. For this particular, I have taken IL-1 example, but this is actually for interferon gamma. For interferons, whatever interferons are there, this LE spot assay or LE spot testing is there in market. So this was not existent before, I would say, year 2005, somewhere these tests came. So these are relatively new. These were not existent in the previous century. And then you can have a whole 96 well plates to study how many cells from different cultures or different environments you might have culture. So here the density is very high, and then it goes up to very low. Uh, so this is some way you can devise your experiments. Another place where antigen antibody interaction come in place is what we call as a flow cytometer. This instrument is flow cytometer. It is comes only very few companies make it, but the most famous company that makes it is BD. BD stands for Becton and Dickinson. We are also BD. We are Mother Dayal Sharma University. These are another successful people. Those are world renowned Becton and Dickinson. Becton Dickinson. So Becton Dickinson makes a whole lot of immunology reagents and equipment. And these equipments are come up because the trust is there on these. There are many other companies which give, but I would say those are price-wise, those are cheaper because people don't have much trust on those equipments and reagents. But then everyone is doing good, and I think it's not कमाना चाह रहा है because earning bread butter is like cutthroat competition out So that is what it is. This is what the setup of flow cytometer looks like. If always you would have double screen, because in one screen you are just looking at the instrument, how your instrument is working, and the other side you are looking at your samples. So you would always have two screens, and this is your equipment. Here you would keep your samples, and then sample is taken up just like a lift, where it goes up and then sample is sucked in. There is whole lot of hydraulic pumps and too much of fluidics in this instrument, and the cost is about nearly 1 crore rupees for this instrument. Other cheaper ones come about 50 lakhs. So that is the thing about it. And this is the working of this instrument. Basically, it's a capillary tube from where the cells would pass through. So nowadays, everything is super fast. Nobody has time. Even scientists working in these experiments, they have cell pass through time of 1 million cells per second. So that is the uh, sensitivity and things that are achieved now, and these are all different types of lasers that focus through on this one single capillary, and at the end it will be able to identify. So it is a cell sorting system. Different types you could instruct the machine. Your CD4 T cells should come here. In this tube, your CD8 cells should come here, and non-T cells will come here. So all the three cells sorting will take place with this instrument. Uh, so this is a little more detail about this flow cytometry, but if you have understood the concept, there is no point in going in much detail. This is the type of graph that are developed. These are called dot plots. So how these are studied, this is one type of antigen over here and other type of antigen on the cell surface. Here is your, this is the master tube where you will see all your cells. All your cells are seen in this. Then you would put a gate on a specific cell type which you are looking. Here it is looking for CD45 positive cell, CD45 positive cell. And let me tell you, CD is a cluster of differentiation, and this cluster of differentiation molecules are there on your lymphocytes. So this neutrophils, T cells, B cells, all lymphocytes would have different types of cluster of differentiation. Today, more than 300 CD molecules are identified, and so um, how sophisticated you are studying that many markers you can use in one go. So he has used CD45, so this basically is capturing all lymphocytes in the blood. That is the total lymphocyte marker, and then they have used a APC staining. Now stains. There are so many. How this flow cytometry is possible? There are many different types of stains that are available in market now. What I was the very first slide where you had plankton and fluorescence. So from there, biotechnologists have picked up all different types of colors. So now the color, because previously there were only three colors: red, green, and violet. Nowadays there are no more era of red and green color. Popularly it was red and green color. Popularly it was red and green color that was used by immunologists in these studies. But nowadays there is no red and green. Nowadays there is a whole spectrum of colors. They have captured the entire range from high ultra red to ultraviolet. All colors have been identified just because of those planktons, and scientists have gone wherever they could find colors. So all sorts of colors are available. Even when this light was off, the our system was on. The world was watching us. Okay. So that's all for our today's talk. Thank you very much for your patience. If you have captured to give a validity? Validity. So before the function ends, Dr. Deepak will give vote of thanks.
Thank you. First of all, I would like to thank the University Authorities for allowing us to conduct this symposium on international immunology. And I also like to thank
Lights
the
on cut है ना भाई, lights.
Immunology Society of India for the free funding to help us to manage the expenses of this symposium. And I also thank my head of the department who has streamlined the things to get this symposium conducted. And I'd like to thank the speakers, Dr. Krishna Mali, who will talk on immunoprotectors, and Dr. Jim Segal from NII, who will talk, and who has planned this symposia and made all the arrangements. I also thank my department, clients and other staff who help us to arranging this symposia. And finally, I'm thankful to the delegates and the students who have attended this conference. Symposia. Without that, there is no meaning to conduct a symposia or conference. So thank you very much. Thanks to all. Thank you.
Not to miss Dr. Chanda, with whose efforts this LT was made available, and our worthy director, sir. We can never miss him because the permission for has been initiated from there. And last 10 days I have been वो पेपर एक जो हमने 4 लाइनें लिखी थी उसमें कि नीड परमिशन फॉर एजुकेशन में इतना सारा कमेंट और कहाँ कहाँ नहीं दौड़ा मैं उसके लिए तो बट इवेंचुअली कल तक हमें परमिशन दी थी yesterday noon when we were testing our system we got the permission ऐसा बिजली लेके आ गया मुझे पता चला था बहुत परेशान हो चुकी थी so with that and if we have missed anybody our Sumit and Babita who were helping us throughout Mr. Deepak आपके लिए तो बहुत जोरदार ताली होनी चाहिए सुबह से मेरे साथ लगे हुए हैं और बॉडी में whenever I was there he was there to help me with everything so and if we have still missed anybody I'm sorry about it and with that I end. Good to कितना होता है ना मेरे ख्याल से अभी ये खत्म हो जाएगा कुछ दिनों बाद तो यू विल डेवलप ई सर्टिफिकेट हमें अब कई जगह पे ई सर्टिफिकेट अपना नाम पता सब कुछ कर दो और कॉन्फ्रेंस कैसी लगी वो भी इसमें फीडबैक दे दो ऑटोमेटिकली जब सबमिट करो अपने आप सर्टिफिकेट सामने आ जाता है तो फिर पता ही सर्टिफिकेट बट ये जो आप लोगों को अभी भी सर्टिफिकेट मेरे ख्याल से 2-4 साल बाद ऐसे Okay, with that we end the Immunology Day program. Thank you very much. Students को देना सर anybody interested in roses and come back आ जाओ जो सबसे पहले स्टेज पे ऊपर आएगा एक ही लेना है भाई